Okay, moving on, I would like to introduce our next speaker who's going to talk to us today about pancreatic cancer research. Let's welcome Dr. Jiyoung Ahn, who is an associate professor in the Departments of Population Health and Environmental Medicine.
Good morning, everyone. I'm a pancreatic cancer researcher. I'm going to share my research, recent evidence of oral health and oral bacteria and pancreatic cancer relationship. As you learned today, pancreatic cancer is very difficult disease. It's very difficult to treat, and over 93% of patients will die within 5 years of diagnosis. It's really sad. And there is no obvious way to screen this disease. So I think it is really important to find out how to prevent this disease. As you learned, risk factors include age, gender, race, family history, inherited genetic syndromes. But these kind of things, we cannot really modify it. But what about risk factors for we can change it? Tobacco use, obesity. If you are smokers and obese, we have more likely to develop pancreatic cancer. So my job is to identify what are the modifiable risk factors for pancreatic cancer prevention. One thing we learned is poor oral health status is related to increased risk of pancreatic cancer. Several years ago, in 2012, we analyzed the CDC based on large national survey data, census survey data. CDC recruited 15,000 healthy individuals, and they surveyed their diet and lifestyle factors. And also they gave a dental survey, so whether people had a poor oral disease, poor inflammation in the gum, or great missing number of teeth. And we looked at these individuals followed over 10 years. And what we learned is people with poor oral health status are more likely to develop pancreatic cancer. For example, people with moderate periodontal disease have 2 times more likely to develop pancreatic cancer. And people with severe periodontal disease are almost 5 times likely, higher risk of pancreatic cancer risk compared to with healthy mouth. So how can we explain this one? It's really, at first I couldn't really believe it. But later, after we published this data, and multiple other studies reported the same thing. For example, 2 other studies conducted in the US reported that same findings. Periodontal disease, if we have inflammation in the gums, and increase the risk of pancreatic cancer. And 1 European study, 1 Japanese study also reported similar things. If you have a greater number of missing tooth, which is indicator of poor oral health, also increased subsequent risk of pancreatic cancer. So how can you really explain this? Well, it seems consistent. Okay, so how can we explain for this? One hypothesis is oral bacteria, because this oral disease is essentially caused by oral bacteria. So we decided to study human bacteria. So let me briefly talk about our body bacteria. In fact, we don't see any microbe in our hand, but in fact, our body is full of bacteria. It's part of our body. We have, research has been said that microbial cells outnumber human cells in human body by 10 to 1. And microbes account for up to 3% of human body. And as you see this pie chart, the study has been done to look at the multiple body site samples. And we learned that each hair, nostril, and skin, and urogenital site, mouth, gastrointestinal site, we all have a different, unique microbe profiles in our body site. So let me define a word. How many have heard of terms microbiome? Yeah, you have read this kind of article in New York Times, and many article media deals with that. So microbiome is a collective term. So micro is a single bacteria, tiny living organism, such as a bacterium, fungus, protozoa, or virus. Microbiome is a collective. So collectively, all the microbe in the human body is the community of microbes. So back to the mouth story. And oral microbiome is a totality of bacteria in the mouth. We now know it's about 700 bacterial species reside in our mouth. And we also know that this bacteria profile is fairly stable over time. We sampled humans from same individuals, oral samples for across the different time point up to 4 to 5 years. And we found that amazingly, each individual bacteria profile is remained the same over time. So why they exist in our body? And they are part of our human body, but they do have actually important functions. They have interact with the immune system, and they metabolize environmental carcinogens. For example, when you smoke, and oral bacteria actually play very important to metabolize, decompose smoking carcinogens. Or when you drink alcohol and oral bacteria, we just begin to understand oral bacteria has some function to metabolize alcohol to aldehyde. And some bacteria is really bad guys, so opportunistic pathogens cause inflammation. And some people, guys, are really good guys, help to digest food components. So I'm a population-based science. My research interest is how across the bacteria share or distribute in the human population. So we looked at the large population of humans, and we surveyed oral bacteria profiles. And what we learned is all humans tend to share the common components. So I bet in this room, everyone shared Firmicutes, Proteobacteria, Bacteroidetes, Actinobacteria, and Fusobacteria. We all shared common members of bacteria. However, I want to point out each individuals have a different quantity. So you make each unique your own signature of bacteria profile. So as you see, this is examples of like 20 healthy individuals of bacteria profile. What you see is like different colors bar chart, right? So it's suggesting there are inter-individual variability. So my bacteria profile is not necessary as the same as Rachel's bacteria profile. We all have differences. So my research question is this inter-individual variability, is different unique individual oral bacteria is related to subsequent development of pancreatic cancer? So I'm going to share my very recent research findings. We just published this paper, Oral Bacteria and Pancreatic Cancer in Gut Journal in last month. So I'm happy to share my recent researches. So we want to study whether oral microbiome, oral bacteria is related to pancreatic cancer. How we studied is we want to study we are lucky to, when 10 years ago, we have recruited healthy individuals, large number of healthy individuals. So we have recruited 75,000 healthy individuals when I was working at NCI. And we also, at the same time, American Cancer Society also recruited a large number of healthy volunteers, 70,000 individuals. So altogether, about 140,000 large number of healthy individuals. And we did a survey, what they eat, what are their lifestyle factors, and then we also asked them to, all the healthy volunteers donated mouthwash samples so we can measure the oral bacteria profiles. And we followed this large number of people over time. So every 2 years, we mailed out the questionnaire. Have you diagnosed any cancer last year? You check. Then we can, and then if they yes, we confirmed their doctors and whether actually it's really get the information. And we followed these individuals over 10 years. Now, after 10 years, among these 140 healthy individuals, 360 people developed pancreatic cancer, but other people didn't develop the pancreatic cancer. So what we study is we look at the microbiome profile when they were healthy, when they donated the samples when they were healthy, and looked at which oral bacteria profiles are subsequently developed, which oral bacteria profile predict for the development of pancreatic cancer. That's how we studied it. So how are we going to study oral bacteria? We haven't really talked about how to measure it. All the days, 5 years ago and 10 years ago, scientists just take the sample and we grow them on the Petri dish. But this method, we now know it didn't really work well because oral bacteria, they grow in the mouth. So if we take out from the mouth, over 80% of human bacteria simply don't grow outside of human body. So with this method, we really cannot study them. And also we can't really quantify who lives what, how much they live. It's just impossible. So scientists came up with excellent idea. Okay, forget about growing bacteria. We take the bacteria samples and we look at the bacterial DNA. So from your basic biology class, the DNA is your own signature. So it's the backbone of the organism. So you can really C, G, A, T, the basic letters. And those letters combine the unique identity for each bacteria. So rather than we growing them, we just look at the bacteria DNA information. So we sequence them. So what we get is largely paralleling these letters. So with this, how we can find out? So we can match this DNA information to fully known bacterial dictionary DNA database. So by this way, we can identify whole bacteria profile, regardless whether they are culturable or non-culturable, comprehensively, accurately. We can characterize the human bacteria, oral bacteria in the mouth. Got it? Okay, so this is what we learned from these 720 individuals. As you see, each oral bacteria is highly structured. But as you see, as I mentioned earlier, we have a very high inter-individual variability. So what the question is any bacteria, oral bacteria, is particularly differ for those people who developed pancreatic cancer or didn't develop pancreatic cancer. So all the complicated computer modeling and complicated analysis, what learn is two bacteria. Is two pathogen bacteria called the P. gingivalus and actinomycetocomitan. So if someone who carry this bacteria has at least 60% or 2 times at least those higher risk of future development of future pancreatic cancer. And these two pathogen bacteria is known to cause inflammation in the gum. So still there is a question, how do oral bacteria influence the pancreatic cancer? I don't have an answer yet, but scientists have shown that oral bacteria actually can spread around the body. We thought it previously, each oral bacteria just affect mouth, but that's not true anymore. So when you have, in particular, gum disease, you have a bleeding gum, and bacteria can it creates like a blood inflammation, bleeding gum, and bacteria can pass through those blood vessels and may travel to your body to travel other organs. Or even though when you have a healthy gum, but when you brush teeth, eating food, is somehow we don't know yet the mechanism, but bacteria can travel to the blood immediately, and create the intrinsic bacteremia state, which the blood can travel. But obviously, we have an excellent immune system. Those bacteria never allowed to circulate all the time. They get cleared. But it looks like there are some mechanisms. Also, think about, we swallow the saliva all the time, and then it may reach to the pancreas. But further study, again, need to be done to underlying mechanism. But one of my collaborators who conducted animal study, and George Miller at NYU. And he conducted actually cute animal experiment. So in the mice, he added bacteria and that trace label, so like a blink, so you can trace that certain bacteria where to move. So he added bacteria at the trace label, radioactive label, and added it to the mouth. And then we were able to show that these bacteria actually can travel to the mice. And then in these genetically susceptible mice, actually developed inflammation and pancreatic cancer, suggesting it's a biological plausibility of oral bacteria and pancreas cancer relationship. So, in conclusion, our study provided the first evidence to show the oral bacteria and pancreatic cancer relationship, and in particular, these two pathogen bacteria, P. gingivalus and actinomycetocomitans. So, so why do we bother to study these oral bacteria? And I think its finding is exciting, but it's more has a great significant implication in the field. First, we can learn about the cause of pancreatic cancer, and if the findings are replicated by other studies, maybe these bacteria may in the future help us to identify people at high risk of pancreatic cancer. Or maybe these bacteria, understanding these bacteria, may lead ways of prevent pancreatic cancer in the future. So what are the next steps? We are currently trying to understand how oral bacteria can reach the pancreatic cancer, and what are the roles of human role, what are human response, what are the bacterial roles in the pancreatic cancer. Many studies need to be done. So in summary for today, we talked about oral disease increase the risk of pancreatic cancer, and we covered what are the oral microbiome, and how can we assay them, and how can you, there are commonality of oral microbiome across the population, but there are high inter-individual variability. And then I showed my recent evidence of possible relationship of oral microbiome with pancreatic cancer. So I will stop here, and we are also recruiting healthy volunteers for our study. So if you are interested in, check this website, NYU Family Study. So I will stop here, and I will take a question after Dr. Leishman's talk. So thank you.